Hello guys, today we have here with us one of the most distinguished researcher and renowned professor of ICT, Dr. Professor Vandana Patravale. I welcome you ma'am on behalf of the Bombay Technologist. We are more than honored to have you here today and I feel truly lucky to have been assigned the task to engage in a surely insightful conversation with you. Before we begin, I would like to throw some light on Patravale ma'am's glorious career. She completed her B Pharm, N Pharm, and PhD Tech from the University of Mumbai and started teaching at ICT in 1991. In her great career spanning over 30 years, she has about 400 scientific publications in national and international journals, 20 patents and 25 more in the pipeline, two trademark registries, 25 book chapters, and two books with international co-authors. She also has innumerous accolades and honors that will take at least an hour to list. She is actively collaborating with researchers as well as industries within the country and abroad and has completed Indo-Swiss, Indo-Japan, and Indo-UK projects. She has executed all major grants from the Indian government focusing on nanotechnology and nanotechnology-based product development. Her research includes development of nanocarriers with major emphasis on malaria, cancer, and neurodegenerative disorders, medical device development, nanodiagnostics, and nanovaccines. She heads the famed VBP research group of ICT, whose students have excelled and stood above everyone else, having done this under the constant guidance and input of Dr. Patravale. She is one of the most beloved and approachable professor at ICT who is more than ready to help her students in any way possible and ensure that they understand the concepts to the core. I once again welcome you, ma'am. And without further ado, let's get on with the interview that we are all so excited about.
Thank you, Karan.
Firstly, we would like to know that with your great skills and expertise, you could have easily had a career in the private sector in any field of your choice. So, why did you choose to be an academician? Was it always your ambition or was it fate?
Fortunately, I had my role model, my father, in my family, right? So Dr. H.L. Bhalla, who was professor of pharmaceutics at Bombay College of Pharmacy.
Okay.
And then later on, he took up directorship at B.V. Patel Pert Center at Ahmedabad. So basically, in my growing years, I've seen him teach, do research, network with people, and do everything, whatever is possible for social cause. So I feel that it's in my genes. Second reason why not private sector, because I wanted to be my own boss. So I think ICT has given me that opportunity.
Absolutely, absolutely. Okay, the name Vandana Patravale is synonymous with research at ICT, with about 400 publications and thousands of citations later. What do you think are the qualities that enable you to achieve such great research portfolio?
Yes, major credit, of course, I'll give to my VBP research group, whom I call it as a family. But yes, if I have to put across the qualities for this portfolio, I would say we always accept any challenging task given to us. So you know that we are collaborating a lot with industry. So anything which is challenging, people would say, no, we do not have facilities, do not have instruments, but we are the first ones to take. So that is first. We do focus on unmet needs, which is very, very important in any kind of research. Of course, passion and drive for research is something which every researcher should have, and that is what we boast of. We do execute all the industry research in timely manner where others fail. So if you're taking any task, you have to commit to it and see to it that you give them deliverables in time. So that is another thing. And of course, we do network a lot with other academicians, other government institutes, as well as private industries. So I think these are the ones which have given us whatever credentials we have today.
Yes, absolutely. And there are some credentials. With your passion and perseverance, VBP is clearly at the top of the ICT research institutions. We all know how successful VBP Research Group is and how every student who has worked with you adores you. And as you said, consider you a family. What are some qualities you look for in a candidate and what values do you try to instill in every student of yours?
So, in fact, as you know, coming from ICT, our mandate is to take in
Right.
meritorious students. So, if you go to see for till master students, we do not have choice to take a specific student. Yes, but for PhD, when we take them on industrial project, we do have choice. But let me tell you, I believe that each student has some positives. So it is role of us as mentors to look at those positives and whatever are negatives to turn those negatives into positives too. So like all fingers are not the same size, students are not the same. So luckily, we do get the best of best students. Let me tell you, even if they come through merit from other places, they are the best. So all we have to do is polish them. But yes, when it concerns, say, some industrial projects, I generally tend to see whether they have the kind of experience which is necessary to execute that project in a timely manner, whether they have good communication skills, interpersonal skills, which are very, very important. Because at ICT, if you have to get something done in a timely manner, it is the interpersonal skills which will matter. We have so many departments, and you need to really talk to the in charges of so many other instruments. So yes, so team culture is what, when we take interview of these students, to shortlist, and you'll be surprised when I tell you that for a single project, typically we get about 200 odd candidates, and we
Wow.
need to interview, shortlist them, interview, and take them in. So first thing which I look at is whether they have their basics clear and whether they can work in a team. Rest of the things can be definitely easily, we can tackle. That's it. And yes, the second part of the question, what values we try and instill? So I would say honesty and integrity. Because believe me, if we have to do technology transfers, which is our prime objective, whatever research we take at ICT, we see to it that technologies are transferred to industry, or the students become entrepreneurs themselves. This will not happen if they are not honest. And secondly, try and tell them that any kind of observation you need to record. That means documentation is very prime. So we need to teach them at times how to maintain records, because it's these small observations that most of the inventions have come in. Whatever patents we've had, it's due to these observations. So many times, they feel that the research has to go in a specific manner, but actually that is not so. Many times you get leads from research which they otherwise would have ignored. I'll give you examples maybe later. Yes, besides that, it's the loyalty part and the family culture. We are very open as a family. We have our lab meets every week. And in those lab meets, in fact, you need to present whatever work you have done in a very transparent manner. Of course, wherever we have signed MOUs with industry, we do not discuss, but the other projects we do discuss in a very transparent manner. So it is required from the other students that they pinpoint wherever they see mistakes, they brainstorm if they are getting stuck. So it's not that always a mentor has all the solutions. I, as a mentor, may not have. Maybe someone else has read something which would be useful here. So openness is something which comes. I think by the time they leave, they definitely have very good presentation skills, as well as integrity and honesty, which I value the most.
Right. So, so much more than the usual classroom learning, the values that you try to instill. That was really amazing. Okay, so you have guided thousands of undergraduates, postgraduates, and PhD candidates till date. But what is the most commendable achievement of a student of yours that you recollect and relish to this date?
Uh, so I feel that actually each student has made me feel proud. As you know, 30 plus years of teaching and research, meaning all my students actually have reached very top positions, be it CEOs, be it senior directors of the company. In academia, you've seen, Dr. Ratnesh Jain, Dr. Prajakta Dandekar, they are my own students. So
Okay.
they are excelling wherever they have gone. So I don't think, I'll be able to pinpoint this. Students have got Gyati Award, which is Gandhi Young Technology Innovation Award from President of India. They have
Wow.
got Veneto Nanotech Award, which is from Italy. Bill and Melinda Gates Awards. So, each of these is special to me. So, and recently, Anjali Pandya from our lab got Commonwealth Split Side PhD, which is also a very prestigious award. So, I feel that it would be really wrong to say and single out something. But yes, something which gives me utmost satisfaction is when my undergraduate students perform well. So you have seen that researchers like say Sayat Mapra and now recently Ekta, Mihika, Sushmit Matre, all of them have contributed to very, very high impact factor publications. So that gives me satisfaction like nothing else.
What's better than that? Not being able to pinpoint on a single accolade. That's truly wonderful. Cool. So the world today is an uncertain place with the pandemic forcing students to learn through the online mode. What do you, as a professor of pharmaceutics, a field based so much on practical knowledge and skill, do to make the most out of online lectures? And what do you think the students to do to reap the benefits of the same?
So to answer this, yes, very correctly you said that the subject which actually I teach, pharmaceutics or advanced pharmaceutics, targeted drug delivery systems, all of them are so very application oriented. So lectures, of course, online lectures, what I try to do is give them theory inputs, but to make the lectures more interesting. Luckily, at ICT, we have student strength, which is less. For masters, it's 6 to 7 students attending at a time, and for BTech, not more than 20, BPharm not more than 30. So we are able to engage them very interactive manner. So you'll be surprised when I tell you that I play Pictionary with them, which is of course curricula based, some quiz, many times Kaun Banega Crorepati. We make teams, actually we make teams and the other teams are supposed to mark these teams if I give a group assignment. So you need to actually look at something different which you can do to keep them interactive. I'm sure all of you would have witnessed that, sitting in one place and just listening monotonous for 5 hours can be really so boring. I have experienced that myself online because I've been attending so many webinars and conferences. So yes, we need to make it interesting. And I resort to all these things which I told you, basically so that I can gauge the analytical thinking capacity of my students. We are just giving, but whether students have really gained that much knowledge which we are trying to give. So for that, we need to figure out what to do. As far as practicals is concerned, what we do is we touch upon very, very quickly, we brush through the theoretical aspect. Luckily, we have made videos of each and every practical. So we show those videos to students so that they understand, even if they do not have a hands-on experience, at least they'll know what is the modality in which they have to go about. And followed by, I show them large-scale videos and even many times characterization studies on videos. So that when they come back, I'm sure, no, this thing is now pandemic is ending and we'll be back soon, and these students will be able to very quickly adapt and do these practicals offline very fast. So these are some things which I do to keep them motivated enough to learn more about the subject.
Right.
And students, what they should be doing, if you ask me, I would say that one thing which they need to do is, listen to us very, very carefully. Believe me, I have seen, and there have been incidences when I call out a name and no one starts their video, but there have been cases where people put on their video and give the right answer also. So it is our sincere request to students that whatever assignments are given to them, they should do it in time. They should be very curious, in fact. They should be proactive. This is actually the time when they can ask n number of questions, to
Right.
teachers and gain knowledge. One student asked, believe me, Karanveer, uh, all other students learn. You've seen, meaning you witnessed that students laugh when some questions are asked by classmates, but that should not be the case. Ideally, you should be very, very proactive. And one thing which I would suggest is, in your spare time, what pharma students at least have to do is go through all the regulatory guidelines. Because if you are conversant with those regulatory guidelines, any industry will take you on a red carpet. They'll give you red carpet treatment. So see that you are well versed with basics. At the same time, you understand the regulations which are there in our industry.
Okay. Combining fun with classes, that is truly the way to go. And surely now we will try and replace Netflix with the regulatory guideline PDFs.
Very soon, you'll be surprised that we are actually coming up with the app which will have such important things taught in a very nice way. So hoping that by next year it could be ready. Yeah.
Wow, looking forward to it. You have wrote numerous amounts of scientific publications, book chapters, and even whole books. But we would like to know where it all began. What was your first publication, and how did you get inspired to write it? Where did you get the idea, and what was your motivation?
Oh my God. Okay. Let me tell you, I'm very, very proud Indian first. Yeah. And I was of the opinion that if we as Indians do not publish in Indian journals, how the standard of these journals will go up. With
Right.
that motto, in fact, my first publication was in Indian drugs. It was my master's work, which was done under the guidance of Dr. S.G. Deshpande, who is, of course, now retired, but still keeps in touch with us. And then I submitted a review on a very recent topic. But to my surprise, that got rejected. And the reason given was that this type of work is not going on in India. That's it, one line sentence. Believe me, it's 30 years back, and today that same work is getting commercialized by each and every industry in India. So, one thing which I would like to say is that as a young scientist, I did not realize that there were bigger sharks just waiting to eat you up. But that did not stop me. That motivated me to write to Elsewhere's journal, no, Elsewhere publication. And believe
Oh,
me, that very publication has more than 900 citations today. So
wow.
that was it, that I started with India. And later on, I had to actually think that I can reach throughout the globe. So once I tasted that, definitely there was no looking back. Then when it came to writing, say, research publications of my own students, in the beginning, I did face challenges, let me tell you, because language of the students was not polished. So I had to do a lot of work. And I still remember a day when I went to, head department of Pharmaceutical Sciences and Technology. At that time, Professor Ackerman, she was there, and I started crying. I said that I have written 5, 6 publications, but they are coming back from editor, reject.
Okay.
He said, don't worry, Vandana, I took 10 years. You're just starting, it's only 1 year. But what I did is then I started reading, the habit which I'm still inculcated, and I try to put it in my students also, that read at least 5 papers a day. Okay. So by reading, what I got is that if it is only because I was a formulation scientist, if I just do formulation and characterization and publish, that's not enough for the journals. They want something additional. So be it say in vitro cell line data, or be it animal studies, or be it clinical studies. So I realized very soon in life that you need to work interdisciplinary to move ahead in life. And that's when I started working with my own colleagues. So Dr. Sadhna Sathe really has been very, very helpful from pharmacology department. Then I started working with textiles. So Dr. Adi Udekar, he started working with us. Chemical engineering. I mean, all departments were there. And then, of course, then I moved out and started working with TIFR also, which has given us many, many publications. So I would say yes, this has been my journey, but there was a time when there was a lag, and that is, you will not believe me, one student who was our undergraduate student. Now he is in University of Florida and University of Hawaii, and will move to Arizona very shortly. His name is Dr. Abhijit Date. He, for some reason, could not go, personal reasons, could not go to US for 1 year. And he was doing research project in our lab at that time. Okay. And we used to have evenings, chatting with each other. I had told him, whatever ideas you have, you should take a notebook and go on writing, because he was full of ideas, let me tell you, full of ideas, and of which many ideas we did execute here at ICT itself. He said, Madam, you're doing such a wonderful work. Why are you not writing your research papers? I said, someone has to take a lead. Unfortunately, at that time, when you're a starter, a beginner, you have, about, at that, you will not believe, I had 24 hours of lectures and practicals. With that, I had students to manage. I had consultancies and industrial projects. So I was not getting time to write. And he took the lead and he started writing my papers. So I think I would give one motivator's name as Dr. Abhijit Date, and the second motivator is Dr. J.B. Joshi, who was our ex-director of UDCity. It so happened that when I went for reader's post now is equivalent to, say, associate professor level. So when I went for that interview, he said, everything is excellent, Vandana, but I don't see your publication number being very high. So at that time, of course, I was ready with the answer. So I told him that, sir, each of my patent is killing 5 my publications. I think all of you should know that unless a patent comes in public domain, no use giving all the details in your publication. So you have to be really careful. And at that point of time, I had 4 patents. I said 20 publications I've killed. But I promise you, I told him that I'll promise you that henceforth I'll give you at least 10 publications per year. And till date, I've kept my promise. There have been always 15 to 20 publications per year from my research group. Oh. So I feel that that was something like a push which was given to me by Professor J.B. Joshi, and I should rightfully acknowledge that too.
Wow, yes. And we see that you have many publications with Joshi sir himself.
Not really, not with Professor J.B. Joshi, but yes, he's been a motivator.
Okay. What a way to kickstart your career and adding up to it every year because of that motivation. That is truly something we adore. You have had a commendable career, as we just discussed, in ICT and more as a student. But what is your most memorable research experience?
Memorable research experience, again, meaning you have sort of pinpointed. There have been again
more
many.
Than one. That's not
so
a
in fact, yeah, there have been many, but yes, I would say 2 cases which I distinctly remember is in research. I told you importance of observations. So one is with a project which we were having sponsored by DBT, Department of Biotechnology, and it was on malaria.
Okay.
And while doing these experiments, we came to know that our placebo formulation, no, placebo is something which does not contain the drug moiety. It has only the base ingredients. Our
Right.
placebo formulation was giving effect which was much more than marketed formulation. Can you beat it? So if student wanted, see, that's where honesty and integrity comes. If student wanted, she would not have reported. I think this
Right.
is a student which you were referring, Karnveer, because her surname is Joshi. So Medha Joshi. So
Right.
I've had many publications with her. Yeah. So basically, she came and said that in animal studies, I have done and I'm getting placebo giving more effect than marketed. I said, don't worry, repeat it. Meaning, rather than saying, how's that possible? Repeat it. Believe me, she repeated in groups of 6 animals, thrives and same results. And that was something which was very important. But because if you have such a platform which already is showing anti-malarial activity, you put any drug in it, it is bound to show you a good activity. And
Right.
much better because the effect either would be additive or synergistic. So that
Mhm.
was a eureka moment. And then, of course, we did a lot of studies with TIFR because later on we took this study to even cerebral malaria. You know, cerebral malaria is a condition where the blood vessels they become so hardened in the brain that they will not allow blood to pass. As a result of it, the patient becomes comatose. So, what is required to treat such a condition is something which can make blood vessels again flexible. And this is a formulation which, by using optical tweezers, the nanostructured lipid carriers, we showed that it actually brings back the flexibility of the vessel.
Wow.
So, and also did a long study to know what is the mechanism. Why is it that this particular formulation is preferentially taken by infected RBCs? It doesn't go to normal RBCs.
Okay.
Very selective targeting to infective ones. So I think that is one. And the second work, which was done by now she's also a faculty in US. Both of them are faculty in US. And Prashita Desai. She came to me, we used to travel together. Generally, with students, I'm very friendly. So she used to come with me till Andheri and then travel till Borivali. So she said, Madam, bad news. Said, what happened? Said, you indicated that form a lipid and see whether it can be put in a amphivallic lipid and see whether it can self-assemble. So I made a lipid, but when I'm purifying, it is frothing. I said, good for you. She said, why good for me? I said, if it is frothing, that means it is having surfactant-like properties. It has surfactant-like properties, meaning it will self-assemble. So
Right.
try whether it is giving you nanosystems. And believe me, it was such a simple formulation. All she had to do is take that lipid, put it in water, and self-assembly has taken.
Wow.
So that is another very simple technology. Later on, of course, we saw that this particular lipid was getting affected by pH, by ions. So it was not something which we could give by oral route. Nasal was okay. Parenteral was okay. That means wherever 7.4 pH is there, it is okay. But as
Mhm.
soon as the pH changes, it is not okay. So
Mhm.
we added one more surfactant to it, and it became then a multiple sort of a self-assembled system, which till today we are using for n number of moieties. It's a very good platform which is available with us. So these two things definitely I cannot forget.
So observation is the key we learn from this experience of yours.
Absolutely.
There
And
are
discussion. Don't just leave it at that.
right, right. Okay. So there are often times in a researcher's life when a lot of people disagree with what you believe in. As you said, many of your publications were not published, but you prove them wrong with your work. You obviously had such experiences. We would love to know some of these.
uh, in this, at least one which I would like to cite is a technology which we have transferred to industry, and currently it is being sold in more than 65 countries. and that is drug-eluting stents.
Right.
1997, when this company people came to me saying that they wanted to coat stents, which are devices, which are used as an alternative to bypass surgery. They open up the vessels so that blood can flow easily. So when they came to me, all over world, people were using bioerodible polymers. So when they came to me and said they want to work on something using laser machine because they were cutting diamonds with laser, first is I had to see what can be done. And secondly, when the stent idea came, I told them, can we use biodegradable polymer? They said, we are okay with it. Any new idea? Because none of them were having any pharma background. They were, as I told you, they were in diamond industry, mechanical engineers at the most, and they had employed some chemical engineers. So anyways, since animal studies had to be done, toxicity studies had to be done, they were ready to experiment. But as I do with my students, on the paper first, I tell them that you convince me why it should work. Then you take up the research. Same way, on paper, I had made a platform to show them why I think biodegradable polymer based coating is much better than bioerodible. Now, bioerodible polymers, when they will erode, right, they can act as locus for thrombi formation. And if thrombus formation takes place, from pharma background you know that if this thrombus goes into brain, there'll be stroke.
Right.
If it gets stuck elsewhere, there'll be other kind of problems. So I said, let the polymer be something which becomes part of the body. And then we gave them this biodegradable polymer based coating, which later on, world over, people resorted to this. We were the first one to get CE mark. Now, being from India, that's something which is very, very important, that we were from India and we got the first CE mark on biodegradable polymer based devices. And we proved all others wrong. Another thing which we did here was what was happening in the marketed devices was, besides these bioerodible, they never used to give the drug at the site, maximal drug. Most of it was getting wasted because for this particular surgery, those of you are aware, from earlier from thigh, the catheter has to reach the place wherever there is a block. Now from shoulder also they are taking, but earlier it was through thighs. That means the tortuous vessels you need to cross. So I gave them another coating. I said, after coating this, you can coat up with the polymer, which is very flexible. So no matter what, nowadays interventional cardiologists are doing it. It's not the cardiac surgeons who are doing. So no matter how they put it, it will still traverse without releasing the drug elsewhere. Releasing the drug elsewhere is a problem. Why? Because on these devices, you are coating anti-cancer drugs, which are cell growth inhibitors. So you don't want it to give side effects. So I think these two things were something which I instantly can think to this question, which changed the total paradigm of drug-eluting stents.
You totally nailed your motivation of making your country proud with such innovations.
That is something for which I've been working, and I'll keep on working till my last breath. Yeah.
Wow. So you have worked extensively on drug delivery systems, especially nanocarriers and lipid delivery systems. These technologies earlier were not considered, like not studied a lot, but these days they have finally become a focus point of industries and are attracting great attention for research. What do you think is the future of drug delivery systems? What is the next big thing?
So yes, drug delivery systems are here to stay, till the mankind, because ailments would happen. And you saw now, right, in pandemic time, the
Yes.
way virus has mutated. So, I mean, it gives us reason to work. Yeah, so I feel the trend now is moving from nano medicines, meaning which was just drug delivery systems containing medicines, to something which is known as nanotheranostics.
Okay.
Now, theranostic is a term which combines two things, therapy and diagnosis.
Okay.
So now we are moving towards, now suppose imagine cancer. We are moving towards technologies which will be able to reach the target and it will image that particular area. One is imaging, so diagnosis is happening. So the patient goes the next time. Clinicians will be able to see what is happening. At the same time, selective targeting also would happen. So, lot of work currently is being put on active targeting. Days are gone when we used to talk about passive. Give anything by IV route, some amount of it will reach the organ. That was what was thought earlier because of the nano size and the leaky vasculature, it will go. But then people move to selective targeting. So if you have, say, glioblastoma, which is in the, of course, the brain, the formulation should release the contents only after it reaches that glioblastoma. So selective targeting. And within selective targeting, lots has happened. People have looked at lipids, polymers, a mixture of lipid polymers, inorganic. But now, currently, it is a cell-based therapy which people are using.
Yes.
So I'm sure you would have heard about something called as nano ghosts. So they are ghost material. Basically, what you're doing is you're taking any membrane which is part of your body. Say take RBCs. Remove the contents of the RBCs from the RBC. So what remains is only a membrane and inject that with your drug.
Right.
So that means now your body has a biocompatible material which is circulating. It could be leukocytes, could be WBCs, could be anything. And then inject. So that definitely you'll have lesser and lesser of side effects and you'll have maximum therapeutic effect. So I would say, talking about futures of wearable devices, first thing.
Mm-hmm.
Devices which can actually detect what are the levels. If you are, say, looking at contraception, then they will actually measure and then inject a certain amount of drug. Okay. This has already happened in case of diabetes patients. They are having, I saw when I visited Spain, people are wearing bands. And what are those bands doing? They are detecting the blood sugar levels, and in response to blood sugar levels, amount would be injected in the body. Self-injection.
Okay.
Okay. So they don't have to do it. That band will do everything. So that is the way technology is moving. And another one is nano robots. Nano? So earlier, right now also, my son is doing neurosurgery now. So he says they are using nano robots not to do surgery. But days not far that when you take a nano robot in, which will act as a pill.
Wow.
It will image. We had written, in fact, the Sanyat Mapara, whom I mentioned earlier, we had written a review on this, wherein already for imaging, there are small cameras which are in these capsules. So the moment you take a capsule, is made up of gelatin or HPMC, which dissolves. And that camera would travel the GI tract. So wherever it goes, it clicks on pictures. Wow. And then it is taken out. You can take it in and take it out. Okay. So there is not far that with this, there will be drugs given. So wherever they find that lesion is there, now suppose colon cancer. If it detects which part of colon it is, the drug would be shot only at that. There is not far. Take it from me. So nano robots is something which I would like to mention. Yes.
Okay. We hope that a few years later, when again you are in a Bombay Technologist interview, these things come true.
They will. And of course, as you have seen, biosimilars are on rise. So yes. We are not talking about, gone are the days when we talk about primary targeting, secondary targeting. It's time for tertiary targeting. If mitochondria has to be targeted, has to be targeted. And nothing should come in the way. So you need to make sure that all the enzymes, everything it is protected from. So drug delivery scene is very, very bright at the moment, I would say.
That is also fascinating. Robots working inside our body. Your research interest also includes vaccine and adjuvants. We recently came across a review in Drug Discovery Today on the prospects of an intranasal vaccine for COVID-19. Can you tell us more about it? And what is your take on how the world can be immunized efficiently with COVID mutant strains on the rise?
Very pertinent question, and that's the reason, in fact, since the first wave, my students have been working at approaches, be it diagnosis, be it therapy, be it vaccines. And we've been very, very fortunate to get even two projects, one on diagnosis and one on vaccines from DBT. So the DBT project actually has come through because prior to COVID, we have worked on Bill Melinda Gates Foundation project, which was on brucellosis, which is also an infectious disease. And what solution we gave there is coming out with intranasal vaccine, which is made using green technology.
Okay.
And the main aim of this green technology is that means it doesn't use any of the organic solvents. And the main highlight of this is that most of the vaccines, as you know, have to be stored in refrigerator.
Yes.
And in fact, why Pfizer vaccine didn't come to India is because it required storage at minus 40
right.
Celsius. So we are trying to make vaccines using technology which do not need cold chain transport at all.
Wow.
So that it can reach to each and every one, including, rural areas. There are, many times it takes 16 to 18 hours to reach a particular village, which are right in the interior. So it shouldn't happen that the efficacy is nil by the time it reaches them. So our purpose is to avoid cold chain transport, to avoid any injection. These can be self-administered, right? Nasal drops can be self-administered. And our hypothesis was that nasal mucosa, the epithelium, is rich in antigen-presenting cells and can take up any antigen and build antibodies in response to that. And plus, we are focusing on something which is very indigenous. So we have already a patent granted in this direction. We are using a muco-adhesive polymer. You'll be surprised. It comes from waste material. What is that waste material? Tamarind seed. So it's a tamarind seed polysaccharide, which has a very good muco-adhesive property. And it is safe to use because in food products it's been used. Plus, we've done a number of celiotoxicity studies. We've done all the histopathological studies to show that the particles which it gives are really very, very safe. So, So what we'll be doing is we are using supercritical technology to encapsulate antigens into this TSP to give us microparticles or nanoparticles, and depends on what we wish to make. Because there was a question which was raised, by a reviewer in one of the papers. Why nano? Why can't you do micro and still get the same results? But yes, nano is definitely giving better results. And what we have done is adjuvant also, which we have used, is totally indigenous. It's a saponin, which comes from Kolaya. So basically using these, we have generated nanoparticles and working on it. Yeah, as regards to your next question, mutants, whether we'll be able to, because they are quickly changing, whether we'll be able to tackle. So for that, if we use the same platform and incorporate genes in it, mRNA vaccine, as you've heard, it would be able to take care, or DNA vaccine, both would be able to take care. So we are still not working on it. Right now, we are working on spike protein. But once you have the platform, anything can be incorporated. So we are sure that we can do that.
Okay. So you are working on drug delivery, literally true.
Yes, it has to be done. I think someone has to take the lead, and we are really fortunate in pharma department. Myself, Professor Padma Devarajan, Professor Vavya, Professor Amin, all of us are working in this area. And to couple that, Professor Prajakta, she's working on biomolecules a lot. So we have a very good team, let me tell you. And given any challenge, we'll be able to tackle.
Yes, that is truly, truly wonderful. Now, we had a great look into your career. Now, let's take a break and focus on some questions about you. Who are the people you look up to the most? Who are your mentors? What are some things you learned from them that stays close to your heart till this date?
I think I'll fail in my duty if I do not acknowledge my parents who have been, I feel they're the role models, both of them. And I have got equal motivation and support from my in-laws. And that's the reason I feel that I am what I am. My father-in-law used to say, "You are our third son." So definitely, I'm indeed lucky to be supported by these. My students, how can I forget them? I think they are the best motivators one can find. And I always tell people, no, that you should be in company of young people so that you become more versatile.
Yes.
ICT, of course, had many role models which I really respect, and that includes our Chancellor, Professor Mashelkar, the earlier directors, Professor M. Sharma, Professor Joshi, Dr. Prasad Yadav. I mean, name each one of them, and all of them have really motivated us. Like, anytime you meet them, you'll get 2 or 3 ideas. So, for example, Professor Sharma, any forum he came, he said, "We do not see any papers coming from on vaccines from ICT. Why is that happening?" Professor Mashelkar, everyone gets Bill and Melinda project. Why ICT is not getting? See, these are small things which actually get, sort of make a mark in your mind, and then you start thinking, why is it? Why is it that no one has written, or is it that no one has got? And believe me, we wrote the project once for Bill Melinda. Second time we got it. Same way, vaccine project came first time itself. I mean, there was no looking back. If you ask me, what do you learn from each one of them? I'll give combined things. That teacher, of course, is forever a learner,
Yes.
right? Uh, Professor Mashelkar has always taught us that nation is a priority. I think this is one thing which none of us should forget, that nation's a priority. We've learned selflessness from him.
Wow.
Staying grounded. Have you seen how simple all of them are?
Yes. Yes.
I mean, they have reached Padma Shri, Padma Bhushan, Padma Vibhushan, and yet they are so very simple. So staying grounded and working consistently, to achieve the goals you have in mind is something which I think I have learned from them. So my parents, my in-laws, and yes, these are names which I cannot forget. And along with that, one name is Professor J.K. Lala, who was principal. He's late Professor Lala, I would say, but he's in my heart forever. Principal of Kim Kundanani College of Pharmacy, from where I did my B Pharm. And he was the one who was responsible for shaping my personality overall, I would say. So it's not only studies which matter, but many more things which do matter. And I think the full credit for the other part, I would give to Professor Lala.
Wow. These are really some things we all must keep close to our heart.
Sure.
You are an academician, a researcher, a consultant, and so much more. But what is it that you love the most about your job and always look forward to?
Uh, so again, these three actually cannot be segregated. They are interrelated. And I think being at ICT has given us a chance to be researcher as well as consultant. Besides, of course, our primary duty is teaching, right? So if you are a researcher and a consultant, whatever experiences you have from there, that you can bring in while teaching. So I feel that yes, teaching is something which I love. I do love. That's my first love. So if I had to pick one, I would select teaching. Research and consultancies anyway come through. So passion is required for research, which definitely everyone at ICT has, and everyone is working consistently. But yeah, picking one thing, it would be teaching.
That simply tells why you are such a great teacher that you are.
I'll tell you what Professor Mashelkar said. I was just looking at some of the saying. He says, education disseminates known knowledge, research converts money into knowledge, innovation converts knowledge into money. Right? So, I mean, if I have to see my institute prospering, right, I need to work on all three. Now that we are a deemed university, I think we need to bring in funds. We have to be self-sufficient. So for that, the other part would be useful. But knowledge has to be disseminated. So I think that is a crux. Yeah.
He really did make an equation combining all these three things.
Sure.
It was wonderful having you, ma'am. Thank you so much. It was so fun getting to know all the wonderful insights of your life and all the little things that shaped your marvelous career. We know this is just the beginning and that you will continue with your glorious research and make our country even prouder.
I feel that, and definitely, for students, I have one thing, our national education policy also says that keep your left brain and right brain in balance. That means you need to be as creative as possible. So on the creative side, I have a flair of writing. So I would like to actually end this by saying something to students, which I have written. It says, अक्स अपने का आईना बनकर देखो, अक्स अपने का आईना बनकर देखो, रूह अपने से कुछ सवाल करके देखो. You asked me that question, no? What students should do.
Yes.
But that they only have the answer. So रूह अपने से कुछ सवाल करके देखो. न मसीहा बनो न ही खुदा बनो तुम, न मसीहा बनो न ही खुदा बनो तुम, हकीकत में जियो, एक अच्छा इंसान बनकर देखो. So I think at the end of it, what we need to do, all of us have to do, is be a very good human being. Everything else is secondary. Research is secondary, I would say. Consultancy is secondary. Teaching is also secondary, but being a good human being is very, very important. So do that, and I'm sure all of you are bound to excel.
That was so beautiful, ma'am. Thank you. We'll all strive to become good human beings like yourself and so many people that have helped ICT and this country prosper. Thank you so much, ma'am.
Thank you.
Thank you so much, ma'am. It really means a lot that you accepted our invitation. And we had a wonderful time listening to you. We couldn't come on the screen, but we were all engaged in what you had to advise us.
Pleasure, Ekta. In fact,